What is photosynthesis? The process in which plant make its own food with the help of CO2, light and chlorophyll. This is the definition you are going to say? No, I'm not saying this definition is not correct, but don't you think that this definition is for the class 9 and 10? Let's try to learn the definition as for the class 11 and 12, isn't it? You see, the moment we talk about the photosynthesis, so photosynthesis is a process in which there is fixation of atmospheric CO2, right? And the energy required is a sunlight, or rather we can say it is an autotropic definition. Because whenever the fixation of CO2 is going to happen, then some source of energy is required. If it is a light, then we call photosynthesis. If it is certain chemicals, we call chemosynthesis. This is Dr. Pankaj Kumar, your mentor for today's session, and in this session we will be talking about photosynthetic pigments. Okay, so first thing first, what is photosynthesis? As I told you, it is a process of fixation of atmospheric CO2, but at the same time we know very well it is a process for it is an oxidation and reduction reactions, right, in which water is going to be oxidized and CO2 is going to be reduced. Okay, and one of the key things for photosynthesis is the photosynthetic pigments. We know very well photosynthesis is divided into two parts, what we call dark reaction and light reaction. Dark reaction is going to occur in case of stroma, and light reaction is going to occur in case of grana. For light reaction, light is required. For dark reaction, it is not that dark is required. For dark reaction, it is that light is not required. So that is the thing as far as the photosynthesis is concerned. Let's talk about the photosynthetic pigments. Okay, you see, the photosynthetic pigments can be divided into three categories, what we call chlorophyll, carotenoids and phycoblins, right? Now let's talk about the different types of chlorophyll. Okay, so it can be chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, E, F, right? And all these chlorophyll are found in chlorophyll A is a universal pigment. Chlorophyll B is found in green algae. Chlorophyll E is found in yellow algae. The typical example is Vaucheria. Chlorophyll F is found in blue-green algae, right? And bacteriochlorophyll, this is the chlorophyll that is going to occur in those bacteria in which oxygen is not released. So an oxygenic photosynthetic is actually being carried out by the bacteria in which there is a bacteriochlorophyll, right? One by one, we will discuss all these things in very detail. So first of all, let's talk about the chlorophyll itself. You know, chlorophyll consists of two parts, right? What they are? One is a porphyrin head and second is a phytol tail. Okay, now what is porphyrin head? Porphyrin head itself is made up of 4 pyrrole ring, or we can also say that is a tetrapyrrole. Okay, so if you say tetrapyrrole, then what happens that magnesium ion is in between the center of the tetrapyrrole, and this magnesium is actually bounded by 2 covalent bonds, right, and 2 coordinate bonds. Mind it, light is very much essential for the biosynthesis of chlorophyll. Okay, for rest of the two pigments like carotenoids and phycoblins, light is not required. For biosynthesis of chlorophyll, light is required. Okay, and this chlorophyll is soluble in fats. Okay, now phytol tail is absent in chlorophyll C. What does it mean? It means that chlorophyll C is of least molecular weight. Many times this question is asked in NEET. So it is of least molecular weight, and bacteriochlorophyll is of highest molecular weight, right? Have a look at the two most important chlorophyll, that is chlorophyll A and B, and how we can differentiate between them. So in front of you, the diagram is of the tetrapyrrole ring, right? You can see there. So in chlorophyll A, what happens that there is a methyl group, okay, and that methyl group is actually present at carbon number 7. You can see there, this is present at the carbon number 7. So in case of chlorophyll A, it is a methyl group. In case of chlorophyll B, it is a aldehyde group. That is the only difference. Rest of the things always remain same, right? This is how, on the basis of molecular structure, we differentiate chlorophyll A and chlorophyll B. Okay. Now let's talk about the carotenoids. Now what is carotenoids? It is also fat soluble, right? Structurally related to, it's a phytol, and their biosynthesis doesn't require light, already I told you. They occur in chloroplast and chromoplast, and molecular formula is C40H56. So these are orange yellow in color, right? There are different types of keratin. For instance, lycopene is a type of keratin that is found in purple sulfur bacteria, tomatoes, and the redness of the red chili is due to keratin itself. So there are three main types of keratin, what we call alpha, beta, and gamma. Okay. So alpha carotene is going to occur in case of dinoflagellates, that is in your syllabus. Beta carotene in case of diatoms, and that is the most common one. And the gamma carotene is going to occur in case of green sulfur bacteria. So these are the few of the carotene that is found, right? Now let's talk about, now you see the chlorophyll extract is prepared in acetone, okay? And what is the fate of beta carotene? Because you see, whatever the green vegetables we eat, there are large number of beta carotene is there. So beta carotene in case of animal is actually getting converted into chlorophyll A. You can see the conversion C40H56. So they actually gets converted into vitamin A. There is one more question. So you know very well that leaf is going to turn yellow in the absence of light. Why? Because what happens in the absence of sunlight, there will be degradation of the chlorophyll, okay? And when the degradation of the chlorophyll is going to happen, what will happen? There will unmasking of the carotenoids, and if the unmasking of the carotenoids is going to happen, we know very well that the color of the carotenoids is yellow, so that's why leaf turns yellow, okay? So that is the reason why the leaf turns yellow in the absence of sunlight. Let's see one last pigment, that is xanthophyll and phycoblins. The xanthophylls are yellow or brown in pigment, that is oxygen derivatives of carotene. So the molecular formula becomes C40H56O2, right? The most common forms are lutein, zeaxanthin, and fucoxanthin. That is the most common form, okay? Now, the moment you talk about the phycoblins, these are blue or red in color. Phycoblins are of three types. What are they? They are phycocyanin, phycoerythrin, and allophycocyanins, and these are the water soluble pigments, right? So that is about xanthophyll and phycoblins. Now let's discuss one more important concept, and that is what is absorption spectra. Pigment, and you know, if we are going to plot it, the plant is going to absorb wavelength light, then we get two peaks, okay? Okay, so photosynthetic is always going to show the amount of light as you can see here. So one peak is in the blue region and another peak in the red region. So that is the important character of any photosynthetic pigments, right? So the total amount of light that is absorbed by the plant vis-a-vis the wavelength of light that is being accepted by them, and if it is plotted in a graph, this is what we call absorption spectra. Now what happens that the absorption spectra of different pigments are different, okay, and there is some range. For instance, we know very well that chlorophyll A and chlorophyll B is going to have two absorption peaks, one in the blue region and second in the red region. So chlorophyll A is going to absorb light less than 450 and more than 650, okay? I am talking about chlorophyll A and B, but even in that they have a very specific range. For instance, the absorption maxima of chlorophyll A is 430 in the blue region and 662 in the red region, okay? For B, the maxima is 453 and 642, right? Similarly, for absorption maxima of carotenoids lies in the range of 425 to 490. That is about the carotenoids. And if you talk about phycoblins, then the phycoblins lies in the range of 500 to 650 nanometer. So just see, below 450 and above 650, red and chlorophyll A and B. In between 450 and 650, then there is the carotene as well as the xanthophylls and the phycoblins, okay? So in that way, you can memorize the range. But from NEET perspective, the exact maxima for chlorophyll A and B is important, and that you have to keep in your mind, right? One last thing, that what is axon spectra? You know, the experiment for the axon spectra was done by Engelmans. Okay, and what he did, he first of all has given an individual wavelength of light to a chlorella, a green algae. Okay, and he has grown the green algae along with the aerobic bacteria, right? And with the help of prism, he broke in the white light into 7 light, and they were given all the 7 lights to the green algae. What he observed, that in the blue region and in the red region, there is the largest concentration of bacteria, as you can see there. So this is the blue region and this is the red region. So maximum concentration of aerobic bacteria is observed, right? What does it signify? It signifies that the absorption peak is in the blue region and the red region. That is, the maximum amount of photosynthesis out of absorbed light is going to occur in case of the blue and the red region. This is how the axon spectra. So the total incoming light that is coming for photosynthesis, only a part is going to be utilized for the photosynthesis, and the part that is utilized, that constitutes the axon spectra. So if you see the axon spectra, it will look like that. So in the blue region it is like that, and in the red region it is like that. So more or less, if we superimpose these things on absorption spectra, it go in hand, because in case of blue also we see a peaks, in case of red also we see a peak, right? This is all as far as absorption and axon spectra is concerned. Okay, so that's all. Thank you. ये कैमरामैन किधर भागा? बंद करो.